All right, I assume everybody can see my screen.
Yes.
Dimitri, so you should start program already.
This should be the presentation mode.
I think you need to shift screens for presentation mode, right? We see your support window right now in PowerPoint. Can you display settings? Perfect. Try and click that one. And then swap presenter views and slideshow, the first alternative.
All right. Is this working now?
No, it's still the support window. Can you try again? Display settings. You're having multiple screens, I presume, right?
No, I just have one screen.
Oh, okay. So that's strange, though. Okay, please try and start the presentation again, then.
Once again.
And then we see what other alternative we'll choose.
All right, so we're sharing.
There's a small button in the lower right corner there. Yeah, that one. Okay, and let's see display. Yeah, now I know. Click that one. Take duplicate slideshow. I think that's the one then. Try that one. Let's see what happens.
This one?
Yeah. Because then you see the same thing on two. Now it works. Perfect.
Yep, now it works well.
Please go ahead.
Okay.
I can maybe make an introduction first, and then I will give the word to you. So thank you for joining us. It's a great pleasure to have you here. I would like to thank you also, everyone, for being here, for being patient with the mess up that we had with the time. So this is part of a seminar series that both our theme, environment and climate, and heritage science organize once a month. In environment and climate, we have a theme that we are very interested in incorporating X-ray techniques and neutron techniques in environmental sciences. So our role is both to network and increase our collaborations, but also to educate the younger people on how they can use X-rays and neutrons in their research. My name is Dimitrios Loutas. I am an associate senior lecturer at the biology department, and I'm the leader of the environment and climate theme. I'm co-hosting today along with Milda.
Hello. So my name is Milda Puchataite, and I'm a researcher at the same biology department, and we are co-leading the environment and climate theme together with Dimitris and a few other people in the Lund University at Links. And I specifically lead the working group 4 that is focusing on studying environmental interfaces using X-rays and neutrons.
And today we have invited Joseph Jakes, who is from, he's a research material engineer at the Forest Product Lab of the US Department of Agriculture at Madison, Wisconsin. His team is focusing particularly on wood cell walls and trying to understand both chemical and structural characteristics of the cell walls. And for this, as part of the research, they also use both X-rays and neutron techniques to get particularly information about chemical and structural characteristics at the micro scale and low, so at the really smaller scales. And with that, I would like to thank you for being here with us today. And now the word is with you. Thank you.
All right. Well, thank you very much for that introduction, and I'll go ahead and get started here. So again, I'm Joseph Jakes with the USDA Forest Service. I always like to start my presentations with the mission of the Forest Service, which is to sustain the health, diversity, and productivity of the nation's forests and grasslands to meet the needs of present and future generations. So one of the major issues with that right now is a lot of motivation for our research is ultimately to improve the forest health. In the United States, we have some issues with catastrophic wildfires. And what causes these catastrophic wildfires is they have an excess of biomass in the forest. When the naturally occurring wildfires come through the forest, what happens is they burn hot and they even kill all the mature trees. So the Forest Service has an intention, to treat about 50 million acres. And so one thing to keep in mind is that a restoration effort, in addition to cost, would also produce about 60 million metric tons of biomass. So to help offset those restoration costs, ultimately the goal is to try to create some sort of market-based value for that woody biomass removed during treatments. And the way that we think about that is, not just finding use for just this low value material, but also just improving and growing the entire forest products industry in general, which of course includes many different products. So kind of my part of that is really to do more basic and fundamental research to address durability issues in wood, to expand wood utilization. So some of those durability issues are fungal decay, fastener corrosion, and also wood adhesive bondline failures. You see that there's a crack right here in this glue lam. And also addressing recalcitrance issues to improve biorefineries. So these woody biomass, of course, made of polymers, and if you can efficiently break down the woody biomass into its monomer components, then you can use those for platform chemicals or biofuels. So in the outline of my talk today, I'm going to briefly just go over what the multi-scale wood structure and also wood-water relationships. I'll just talk briefly about kind of what led me to synchrotron X-ray research and how I got there. And then I'm going to provide a few snapshots of how we use synchrotron-based techniques, including X-ray fluorescence microscopy, X-ray absorption near-edge spectroscopy, X-ray computer tomography, and SACS, small angle X-ray scattering to study wood. I'd just like to point out that as a U.S. government employee, all my publications are freely available. You can either get those from the Forest Products Laboratory website, or if you go to something like Google Scholar and search me, you can usually find the links to the PDFs back on the FPL website from Google Scholar. And then the majority of my talk today, we talk about different sample preparation strategies that we developed for studying wood and synchrotrons. So for those not familiar, there's multi-scale wood structure here. There's two main types of trees. There's softwoods. Those are the coniferous pine trees, and then hardwoods, which are deciduous trees like oaks and maples. One thing to keep in mind when studying wood always is that wood was designed by the tree to solve tree problems. So those primary tree problems in a living tree are mechanical support, so maintaining the tree above ground, liquid conduction, so moving water from the roots up to the shoots, and then food produced in the leaves back down to the roots, and also synthesis and storage of biochemicals. So both softwood and hardwood, if you look at their stem material, you'll see these concentric growth rings, and so this is the longitudinal direction up and down, and the growth rings define the other primary planes of wood. So the cross section is a transverse section. The direction from the pith out to the bark, that's the radial direction, and defines the radial longitudinal section. And then also the tangential to those growth rings is the tangential longitudinal section. If you look at the cellular structure of wood, there's another difference there between softwoods and hardwoods. Softwood is a simpler cellular structure where the primary type of cell is called a tracheid, and the tracheid is responsible for both mechanical support as well as liquid conduction. In softwood, there's two different types of cells. There's the vessel cell, which has a larger opening and is probably responsible for that liquid conduction of water from the roots to the shoots. And then the fiber cells, they're smaller and have smaller lumina. Those are primarily designed to help for mechanical support. And then both types would have these ray cells that run from the pith out to the bark. Now, if we go in and look at smaller length scales, both the fibers and tracheids have a similar cell wall structure. So fibers and tracheids, they have these empty lumina, which are just empty spaces. Then S1, S2, S3, these are the secondary cell walls, which are essentially these nanofiber reinforced composites, where you have cellulose nanofibrils embedded in matrix of hemicelluloses, which for softwood is typically glucomannan and xylan, and then also lignin, which is an aromatic kind of encrusting polymer. And then these individual cells are held together by a lignin-rich material called the compound metal lamella, which is primarily lignin with a little bit of hemicelluloses. So the amorphous cellulose, hemicelluloses, and lignin, although they're all polymeric materials that have a lot of hydroxyl groups, so that makes wood a hygroscopic material. I'd like to point out that in wood, moisture can exist in generally in two different types of states. One state is what's referred to as free water, which is basically liquid water that's formed in like the lumina of wood. And then the other state is bound water, which is water that's actually being held by the intermolecular attractions within the wood polymeric cell wall structure. And so The amount of water in wood is defined as the moisture content, which is the mass of water divided by the oven dry mass of the wood. And as you condition wood to different to different environmental conditions at higher humidities, it absorbs water. So up to what's called the fiber saturation point, which is about 30% moisture content, all of the water in wood is in its bound state. And so it's inside of the wood cell wall polymers, which is causing plasticization and really affecting a lot of the properties that we're interested in measuring, such as mechanical properties, swelling properties, mass transport properties. And I'll show later how we actually build humidity chambers in the synchrotron beam lines to actually study these properties over different levels of moisture content. And then once you get above the fiber saturation point, that's when you start to get free water forming in wood. So just briefly, kind of what led me to synchrotron X-ray research. Back in 2004, I got an internship here at the Forest Products Laboratory, and I spent an entire summer making and breaking wood adhesive compression shear blocks. I did that thousands of shear blocks, and then apparently I did a good job because they decided to hire me on into a student training program at the Forest Products Laboratory. And then from 2005 to 10, I was a PhD student. I was working here at the Forest Products Laboratory, but I was doing my PhD studies with a professor in the material science program at the University of Wisconsin-Madison. And my entire PhD was spent developing nanonotation techniques to actually assess mechanical properties of wood cell walls. So that included developing a method to prepare surfaces without any sort of embedment. See, they got empty lumina and very smooth cell walls that we can place these nanotations. These nanotations are always near heterophrase interfaces and free edges, such as an empty lumen. So I developed methods to account for those edge effects. And then because nanotation was developed to test metals and ceramics, and of course wood cell walls are polymeric materials, I also did a lot of work understanding what these nanotation measurements mean in polymeric materials like wood cell walls. Then in 2010, I graduated from my PhD and became a research materials engineer here at the Forest Products Laboratory. And I made it my goal to become a material scientist of the wood cell wall. So material science is, of course, studying the relationships between processing, structure, properties, and performance. So I was able to measure mechanical properties, but I wasn't able to do other types of properties and also understand different effects of processing. One example is I was interested in understanding wood adhesive bond lines and how the adhesives interact with wood cell walls right at that bond line. So I was able to do these nanotation measurements where I could test a cell wall next to an adhesive. This is a phenylformaldehyde filled lumen. And I could find that the cell wall next to the adhesive had higher mechanical properties than the cell walls away from the adhesive. But I didn't know, how much adhesive has to infiltrate in the cell wall to cause those changes in mechanical properties. And those are the types of basic questions we were interested in. So really, I need a technique to be able to quantify adhesive filtration on the same length scales as my nano notations. It happened in 2012 that I had the opportunity to visit the Advanced Photon Source at Oregon National Lab. And there I met a beamline scientist who suggested that we try X-ray fluorescence microscopy to actually map a bromine labeled adhesive. And you can see here this image here that I'll talk a little bit more about later, that we did have success. We were able to take a bromine labeled phenylformaldehyde and glue pieces of wood together, then map the bromine to be able to tell us how much adhesive is actually going into those wood cell walls. All right, next I'll get into a couple of snapshots of the research that we've done. So the four techniques I'm going to touch on here are X-ray fluorescence microscopy, or XFM, which is used to map and quantify trace elements in wood. X-ray absorption near spectroscopy, or XANES, which is used to assess the oxidation states of those elements. X-ray computer tomography, or XCT, which reveals the three-dimensional structure of wood with sub-micrometer spatial resolution. And then also some small angle X-ray scattering, or SAX, which probes the nanostructures of wood. And all these experiments were done at the Advanced Photon Source, located in Oregon, Illinois, which is actually, it's about a 2 or 3 hour drive from where I'm at here in Madison. So it's quite convenient for me to go down there and do experiments. I'd also like to point out that from 2013 to 15, I had the opportunity to actually be a resident user down there. So that's really when we were able to push and get a lot of these research programs going. So the first snapshot here, wood adhesive bond lines. So again, as I mentioned, I was interested in understanding how much adhesive infiltrates into wood cell walls. So we did these different experiments using a bromine labeled phenol-formaldehyde adhesive. So we used able to use XFM to map how much adhesive goes into the cell walls themselves. See some of these luminaire filled. Another advantage of using bromine is that it increases the density of the adhesive. So when you do XCT, it increases the contrast between the wood cell walls and the adhesive. And so you're able to computationally remove the wood structure and actually see the 3D structure of the adhesive flowing into the wood structure. It's like down the ray cells and up trachea lumina away from the bond line. And then another advantage of synchrotron X-rays is that, of course, X-rays are penetrating nature, so you can build, steps of humidity chambers, and also the beamlines themselves are relatively open. So you're actually able to incorporate these different humidity chambers into the beamlines themselves. So here's just an example of a humidity chamber that we built for an XCT beamline, and this is just a cross ply adhesive bond line. We have the longitudinal direction that doesn't swell hardly at all, whereas the tangential swell is about 10%. And you can see that as you cycle between dry air and 98% relative humidity, you're able to see how the wood bond line swells and shrinks. And you can see you got these micro cracks forming in the bond line. Another really interesting study that we were able to do at the synchrotron was we had a theory about how larger diffusants, such as mineral ions or small organic molecules, we had a theory that they only diffused into a wood cell wall at elevated humidities, elevated moisture contents, but we had no way to actually test this theory. And so what we did was we were able to devise an experiment at the beamline where we took a 2 micron thick section of wood, and we were able to deposit some mineral ions. In this case, it was copper from a copper sulfate solution, and then we built a humidity chamber that fit into the beamline. So this is the detector here, and the beam is coming in from behind here, and we're actually able to map this implanted copper in the section. And you see that under low humidities we saw no diffusion, but then we got the high humidities. We're able to see obvious diffusion of the copper through the cell walls. So we repeated this experiment, different wood orientations, different mineral ions, different tagged organic molecules, and we always saw that at low humidities there was no diffusion, and at higher humidities there was diffusion. And we were able to combine these experiments with some nanonotation-based dynamic mechanical analysis to actually prove that what's actually controlling diffusion through wood cell walls is a moisture-induced flash transition of the amorphous polysaccharides inside the cell wall. We've also found that these techniques are great for studying the distributions of physiological relevant trace elements in fungally decayed wood. So once the experience we do these soil bottle tests where we expose a wood block to a specific fungus inside of a soil bottle. They have so here's just an example of the calcium and iron maps. So you see that on decayed wood, you have kind of a relatively even distribution of calcium and basically no iron. But then after exposed to Deuterium, which is a brown rot fungi, you can see that the calcium is being controlled by the fungus, and also that there's being iron translocated from the soil up to the wood block. So these large field of view maps, they don't tell you where these elements are in like the fungal hyphae or the cell wall themselves. So we can also go in and cut thin sections from different regions in the wood blocks, and then you're actually able to look at the cell wall distributions. So you see that in the undecayed wood, there's more calcium in the middle lamella than in the secondary cell wall, and then through different stages of decay, you see that it's being changed, and then you can see that the iron initially is accumulated in the middle lamella, and then it's kind of throughout the cell wall later in the later stages of decay. We're also able to do X-ray fluorescence microscopy tomography. So in this case, this is a wood section with some dried extracellular matrix on it, and you're able to see that you can map all these different elements at the same time and see their relative distributions also in a 3D space with sub-micrometer spatial resolution. Another snapshot here. This was with some collaborators at the National Renewable Energy Laboratory in the United States. and they were interested in trying to get woody biomass to accumulate iron in the cell walls to act as a cold catalyst during the deconstruction of lignocellulosic biomass. And they had these transgenic Rhapsodopsis specimens, and they were able to see that within the total stem there was more iron, but they couldn't tell whether that iron was in the cell wall or not. So again, we were able to use XFM to show that indeed in the transgenic Rhapsodopsis that there was more iron in the cell wall itself. Another one I have my colleague here at the Force Pox Laboratory, Sam Slinka, was really interested in understanding metal fastener corrosion in wood. And in particular, we had in the United States, we have different types of copper treated wood, and the different copper treatments for showing different behavior with regards to fastener corrosion. And so one thing that Sam was interested in was, where's the copper reduction taking place? So in this case, these are sections of wood that were cut from a metal fastener corrosion study, and this is where the iron fastener was to begin with. You see that indeed, iron is diffusion into the first couple layers of cell walls, and but then if you test the oxidation state of the copper, within this corrosion zone versus in the control zone, you see that there's no difference in the copper oxidation state, which is telling us that the reduction is likely taking place right at the fastener surface. Another snapshot here was I was fortunate to have Magdalena Broda, who was here at FPL as a Fulbright scholar. She's from the University of Poznan in Poland, and she's interested in developing new types of conservation treatments for waterlogged archaeological wood based on organosilicons. So she was treating different types of degraded wood with different types of organosilicons, and we were able to use XFM to actually show how much of those organosilicons were actually infiltrating and entering into the wood cell walls. This is a small study that we did. Look at using microprobe sacks to look at different wood cell wall layers. Again, we built a humidity chamber to actually fit inside of the beam line, and then we can actually able to probe different cell wall layers. And so in the secondary cell wall, you kind of this fiber diffraction peak. There's a scattering pattern, whereas if you look at the complement lamella, that's more of an amorphous type of structure. We did some preliminary experiments with this. At the time, I had a graduate student we also did some neutron scattering experiments, and the neutron scattering experiments were easier to interpret at the time. So we pursued the neutron scattering, and we haven't done much with this since then. And then the final snapshot here is looking at different types, looking at how the wood cellular structure and wood cell wall actually swells with moisture in the same piece of wood. So we're able to do these XAT experiments again, where we test the same piece of wood at different humidity levels, and we can segment the XAT image to look at how the cell wall swells, and then by looking at the outlines, you see how the cellular structure swells, and that allows us to make the comparisons between those two types of swelling within the multi-scale wood structure. All right, so now getting on to the main purpose of this presentation, I'm talking about how we can actually prepare wood samples for doing these types of experiments. So a lot of these high resolution images that I showed were done using 2 micron thick wood sections for wood. And so the way that we cut those wood sections is we use a diamond knife fitted to an ultramicrotome. I'd like to point out that we developed all these methods with no type of sample embedment, and also we just cut these specimens under ambient conditions. So that makes it easier, because oftentimes these synchrotron experiments are also conducted under ambient conditions. So the first step is to take a block of wood, and then we just use a hand razor to kind of create a blunt apex in the region of interest. And then we fit that into the ultramicrotome with a diamond knife. So typically we use a 35-degree diamond knife, whenever possible, because it's more durable. But if you have a highly degraded sample or a sample with really thin cell walls, like those Rhapsodopsis specimens, then we'll use a 25-degree knife. Some of the tools that we use, these are pretty simple tools. They're just wood sticks where we shape the ends in different ways. So we have got a spatula stick, a pointy stick, then eyelash tool, which is just a sound. It's one of your eyelashes glued to the end of a stick. This is a very, very important tool when dealing with these thin sections. And then, we can cut these sections in different orientations. So we have the transverse sections where you have a cross-section of those cells. Then we also can cut tangential longitudinal sections, which go along the longitudinal direction. We typically do tangential because then you're doing cross-sections of those ray cells. If you try to do tangential radial, then inevitably you end up with a ray running through the middle of your section and it just falls apart when it's this thin. Some strategies to kind of minimize this curling. One thing you can do is you can cut smaller sections. If you do end up with curled sections, one thing that we've done that works pretty well is you just take a scalpel and just kind of cut this in half. Then you have some smaller sections that are not as curled. But oftentimes what we end up having to do is, as you're actually cutting the specimen, you actually just take your eyelash tool and just hold it flat as it's being cut. And if you use the slowest speed on the ultramicrotome, this is it's fairly easy to do using the optics of the microtome. All right, so once we get these different sections cut, they'll be sitting here on the knife edge, and then I take a eyelash tool and just gently pick up the sections, and I place them in these small plastic petri dishes. And then once you have your sections in there, you just put the cover on, and then these can kind of sit indefinitely until you're ready to mount them for the experiments. So some of the first set of experiments I did using XFM on the adhesive bond lines, we actually use these foldable copper TM grids. So you have to cut a long section and then manipulate it under a dissecting microscope to get it to and then fold it and clamp it inside of these foldable TM grids. One thing about these is that the sections were not always flat. You can see here in this optical microscopy image. Of course, sections need to be long enough to actually span the hole. And also with XFM, if you're using a copper TM grid, you're always going to have some copper contamination issues. And so the way that we do it now, kind of for standard XFM imaging, is we actually sandwich wood sections between these silicon nitride windows. And so these silicon nitride windows, they come in different sizes, but the one we typically use is has a silicon frame, that's 5 millimeters on a side and 0.2 millimeters thick. And then the silicon nitride window is typically this 2 by 2 millimeters and then 200 nanometers thick. One thing to know with these silicon nitride windows is that the film itself is always flush with one face. So in this particular picture, you can see the bevel here. So it's actually flush with the bottom of the frame. So this is an aluminum stick that fits into the beam line, and on the top of here is a sandwich that's already made. I'll just kind of go through the steps of how we make that sandwich. So first, to mount the silicon nitride window, we use 4 small drops of typically just a clear nail polish. And then you put the first window down such that the flush end is up facing you. And then you carefully use an eyelash tool to transfer your sections from your plastic petri dish onto the silicon nitride window. And if you try, and in my experience, using anything besides a eyelash tool to try to manipulate or move these sections causes that silicon nitride window to break. And then once you have your sections, carefully again put 4 more drops of nail polish down, and then go ahead and put that second silicon nitride window on top. In this case, the flush slide down, so it presses the sections flat. So here's just an example of one of these silicon nitride window sandwiches loaded up with different wood sections. One great thing is you can mount lots of samples, and the samples are held fairly flat. A couple disadvantages here is that it can be difficult to see silicon because of course you're getting a large silicon signal from the silicon nitride window. You can have some absorption issues for the lower K elements because those fluorescent photons, they need to actually pass through that 200 nanometer silicon nitride material before it gets to the detector. And of course you cannot do humidity experiments because they're being held between those 2 windows. So for doing humidity experiments, what we devise is we have to have longer sections, and then we tape them over a slot cut into a capped on film. So to make the small pieces of tape, the trick here is we just take a plastic Petri dish, put a piece of capped on tape on it, and then you can actually just cut little pieces with a scalpel or razor blade, and then use your wood scalpel tool, or sorry, wood spatula tool or fine tweezers to pick off little pieces. And then we just cut the slot in the capped on film just by using a scalpel by hand. So you'll have your small wood section. And then you take a piece of tape on the end of a spatula stick and just kind of touch it down to one end, and then it's attached. And then you transfer that to your slot on your piece of capped on film. As I mentioned, we do this under a dissecting microscope to help us see everything well. And then you just use your pointy stick to press the tape down onto the capped on film. And then to stretch it across the slot, you just take another piece of a small piece of capped on tape and just kind of catch the end, and the tape just grips it and holds it. And you just kind of pull it gently and use your pointy stick again to press the tape down on the other side. And then you have your section that you're able to do your experiments with. So for a little bit larger samples, something around about 0.1 millimeters, a tool that we typically use is called a sled microtome. So these are common in wood science laboratories for sections for our optical microscopy. So the basics of what the sled microtome is you have your blade attached on a sled that you pull forward manually, and then your specimen is mounted, and this clamp here, and then this whole clamping mechanism actually moves up this slope right here, and by using this crank. And so one crank is 1 micron. So one click is 1 micron. So if you do 100 clicks, that moves the specimen up 100 microns. And then once it's in place, you just pull the sled forward to actually cut the section. So you have a section that's whatever thickness you want. I think this one was 100 microns. And then once you have these sections, I again go to a dissecting microscope, and the octastriate sections curl a bit. So typically I just use glass slides to kind of hold them flat, and also as a straight edge, and then just use a hand razor to cut small pieces. And then once you have these small slivers of wood, there's different ways to mount them for your experiment depending on your beamline and the mounting requirements. Some ways that we've done this in the past is take one of these slivers of wood and first actually just tape it to a small glass tube, which was just breaking off a piece of a disposable pipette tip, and then take that piece of glass and actually tape it or glue it to the aluminum stick that fits into the beamline. Another way you can do this is if you have a large enough piece of wood, you can just bond it directly to the aluminum stick. But we should always make sure we use a viscous adhesive, such as like a 5-minute epoxy, because you don't want that. The adhesive needs to be viscous enough such that it doesn't wick to the region of interest. And then when you go to even larger samples, the preferred method really depends on the direction of the cut with respect to the anisotropic cellular structure. So again, remember that you have the longitudinal direction and then also the transverse direction. If you're cutting longitudinally, it's actually really easy to fracture wood when you're cutting in this direction. And so to make kind of specimens larger than about 0.1 millimeters, we use this custom built razor blade guillotine a lot. So this is just on a press, and you can just move the handle and it pushes the razor blade down into your specimen. So it actually works pretty well. Just line up your specimen and then press the razor blade into it, and it's less of a cutting motion and more of just like a very carefully directed cracking motion. So it's more of a fracture than a cut. But given the structure of wood, it works really well for making specimens this size. And if you want to cut the transverse direction, I would typically just use these a very fine tooth saw and a small miter box. That's just something that you can typically just get at like a hobby shop. And then to mount these larger specimens, for micro XCT, we've oftentimes just used clay, where you just take a small amount of clay and put your wood section in there and hold it. But we are finding that as synchrotron experiments get faster and faster, and you can even do like real-time XCT imaging now, sometimes you get some vibration issues when you with the clay. And so going forward, we're going to always use mechanical clamping, some sort of mechanical clamping, either like a pin vise or just a small clamp, just to hold the specimen in place. Then for experiments like large field of view XFM, it depends whether or not the edges are important. If your edges are not important, then you can just glue it directly to, these are aluminum frames that fit into that beam line. But if your edges are important, one thing that we've done is we've actually just used ultralean film to kind of make little baggies where we can suspend the specimens, and then they were able to reliably measure also the edges in addition to the centers of the specimens. So coming to a summary, synchrotron X-ray research is impacting many areas of wood science research. From wood-adhesive bondlines to diffusion and wood cell wall layers, fungal decay, linear cellulose feedstocks for biorefineries, metal fastener corrosion, conservation of waterlogged archaeological wood, swallowing in wood cellular structure. And I think there's other opportunities as well. And I think as I showed, sample preparation often requires some creativity, practice, and time. And also when you're thinking about sample preparation in these humidity chambers, you always want to be the photon as well, because if the photon can't get to your material, and if the photon can't get to the detector, then you can't really do experiments. So you guys should be thinking about the path of the photons. With that, I'd like to thank everybody for your attention, and I'd also like to recognize a lot of my collaborators over the years, both at the Force Fox Laboratory as well as Advanced Photon Source. These are all the different experiments, different beam lines that we've done work at the Advanced Photon Source.